Cellular respiration is the process through which humans take chemical energy and convert it into the form of ATP. Through cellular respiration, a total of 36 ATP are produced. Carbohydrates, fats, and proteins can all be used as fuels in cellular respiration, but glucose is the most commonly used example and is the one we will be using today. Cellular respiration consists of 4 stages: glycolysis, pyruvate oxidation, Krebs cycle, and the electron transport chain. First up is glycolysis. Glycolysis takes place in the cytoplasm of the cell and is an anaerobic process, which means it does not require oxygen for this set of reactions. As we will see, this stage is not very efficient as only 2 ATP are produced. The rest will be released in the next several stages. First, 2 phosphate groups are transferred to glucose via kinase enzymes, where ATP is converted to ADP, and fructose 1,6-bisphosphate is produced. This sugar then splits into one DHAP and one G3P molecule. The dihydroxyacetone phosphate, or DHAP, is further converted to G3P, which leaves us with 2 G3P molecules. Each of these G3Ps undergoes an oxidation process, which in turn reduces NAD+ to NADH. The oxidized G3P then bonds to the inorganic phosphate and becomes 1,3-bisphosphoglycerate, or 1,3-BPG. Next, our ADP bonds to the phosphate to make ATP, which leaves us with 3PG. The phosphate transferred to the second carbon, which makes it 2PG, which then loses water, producing PEP, or phosphoenolpyruvate. This PEP is finally reacted with ADP to give us ATP and pyruvate. Remember that this whole process occurred with each of our G3P molecules, so in total we are left with 2 ATP and 2 pyruvate molecules. After that, we have 2 pyruvate molecules entering the mitochondria to be oxidized. One carbon dioxide atom is removed via decarboxylation, and then 2 protons are removed using NAD+. An enzyme called enzyme A attaches to the remaining carbon atoms, which creates 2 acetyl-CoA molecules, ready to enter the Krebs cycle. Once acetyl-CoA enters Krebs cycle, it joins with oxaloacetate, which creates a 6-carbon citrate. This cycle consists of 8 reactions, each of which breaks down the citrate until it reaches the form of oxaloacetate once again, which cycles back into the same cycle. This cycle releases carbon dioxide as a byproduct and creates 2 molecules of ATP, but most importantly, 6 NADH and 2 FADH2 molecules are formed, which will be used in the electron transport chain to extract more energy. This occurs in the inner mitochondrial membrane. First, the NADH we have produced in glycolysis enters the mitochondrial matrix and is converted to the form of FADH2. Also, the pyruvate from glycolysis enters the matrix and goes through NADH dehydrogenase, which reduces the NADH and forces the protons outside of the membrane to create a proton gradient. Now the FADH2 and NADH within the matrix are transported through the cytochrome BC1 complex via ubiquinone, where once again the electrons pass through the next complex called the cytochrome oxidase complex. And finally, because of this electron gradient that occurs, protons are controllably channeled through the proton channel, which allows ADP and organic phosphate to create ATP. For each FADH2, 2 ATP were created, and 3 were created for each NADH, which totals to 32 ATP. That we have obtained our grand total of 36 ATP from one molecule of glucose. Now we're going to look at photosynthesis. This is the process by which plants create energy, and is created out of 2 main processes, the light cycle and the dark cycle, which is also called the Calvin cycle. Here we have the light reactions. In this set of reactions, light is used to create ATP in the chloroplast of the plant cell. First, the photon of light hits the antenna complex on photosystem 2, which excites 1 electron to a higher energy level. This is called photoexcitation. This electron jumps from one complex to another until it reaches the PEA, or primary electron acceptor. The PEA is then reduced, and this process is repeated until we have 2 electrons at the PEA. These 2 electrons are transported through the electron transport chain via plastiquinone and to photosystem 1. These electrons are then moved to NADP reductase via ferredoxin. and then NADP plus is reduced to NADPH using hydrogen ions from the stroma. At this point, we have a high concentration of H positive ions inside the membrane, which are allowed to exit via ATPase. In the Calvin cycle, we use carbon dioxide through a series of reactions to create G3P. One of the molecules of G3P goes towards the formation of glucose. The rest of the molecules go through the sugar shuffle, which recycles into RUBP, which then bonds to carbon dioxide and goes through the cycle once again.